Emerson, currently a postdoctoral fellow in TPU-HSC, Texas, in the United States. But in this presentation, I'm going to tell you part of my PhD thesis that I did in Valparaiso, Chile. There, I studied the voltage-gated proton channel HV1. This is a protein present in the membrane of cells, which function is to dissipate intracellular proton accumulations. This function is a consequence of the biophysical properties of the channel. HV1 is voltage-dependent, pH-dependent, and highly selective to protons. You can see that the same structural domain contains a voltage sensor and a permission pathway in this dimeric protein. Actually, if we delete the N and C-terminal domain, monomeric channels are produced, which maintains the biophysical properties of the channel. This is a simple model to explore how the channel functions. In particular, we would like to know if the activation of the voltage sensor and the opening of the permission pathway are concerted processes or they occur in a series of steps. To answer these questions, we can study the voltage sensor movements. Gating currents are a direct measurement of this movement. They are transient currents produced by the displacement of charges on the voltage sensor during activation and deactivation. As you can see, these currents have been measured for different channels. However, when I started my thesis, there were no recordings of HV1 gating currents. Gating currents are small, so we have to measure them from many channels. To do that, we express the Ciona intestinalis HV1 channel in Xenopus oocytes and study the currents produced by patch clamp in excised patches of membrane. You can see that the monomeric channel produced voltage-dependent proton currents, but there was an initial component of current also. However, this component is contaminated with proton currents. We decreased them using a low conducting channel. In this case, gating currents are observed at the beginning, followed by small proton currents. We feed the traces to this equation to separate gating currents from ionic currents. Then, the blue traces showed isolated gating currents. These isolated gating currents showed a voltage-dependent kinetics of decay, which is quantified here, and also the charge displaced saturated at high voltages, which is quantified in the QV curve. These are two hallmarks of gating currents. However, our recordings suggest that not all the gating charge displaced during activation came back during deactivation, which is incompatible with gating currents. Using an on-recovery protocol, we found that the off-gating currents cannot be detected because they are too slow. Despite this, all the gating charge returns, as you can see from the magnitude of the feed and the initial on-gating currents. This result demonstrated that the current transient produced are truly gating currents. Then, we activate the voltage sensor with a 200 mV depolarization preceded by a variable prepulse. You can see that the on-gating current of the second depolarization changed completely as the prepulse increased. This can be explained by the existence of multiple steps during activation. At higher prepulses, the voltage sensor activates from states closer to the active state, altering the on-gating current's kinetics. If this is correct, we should observe a similar effect on proton currents from the conducting channel. Actually, this was the case. The delay of proton currents changed at different prepulses, which is known as a Kolmogg shift. Similar to what happened with gating currents, this phenomenon is caused by the existence of multiple closed states preceding the open state. Interestingly, in this recording, we can observe the effect in both the ionic current and the gating current as well, which you can see at the beginning of the pulse. All these results indicate that the voltage sensor activates in various steps, and therefore the activation of the voltage sensor and the opening of the permission pathway are not concerted processes, even though they occur in the same structural domain in HV1. To finish, I would like to recognize the amazing biophysicists I have the pleasure to meet and work with during my PhD. They are Ramón Latorre, Osvaldo Álvarez, Alan Neely, and my thesis advisor, Carlos González. Thank you guys, and thank you for watching.